Today, the topic I have selected is liposomes as a potential drug delivery system. The part 1, the topic I have selected is been covered in B.Pharm 7th semester and also it has been covered in M.Pharm 1st semester and 2nd semester. The content I will be discussing in these slides are introduction, structure of liposomes, composition of liposomes, mechanism of liposome formation, advantages and its disadvantages, classification of liposomes and method of preparations. Starting with introductions, firstly I will discuss what are liposomes. As we know, liposomes are simple microscopic vesicles in which aqueous volume is entirely enclosed by membrane composed of lipid molecules. Liposomes are concentric bilayered vesicles in which aqueous volume is enclosed by lipid bilayers mainly composed of natural or synthetic phospholipids. Liposomes is a Greek word which means lipo, fat, and somes means body, and it was first produced by Alec D. Bangham in 1961 in England. As we are seeing the design of liposomes, the liposomes are being formed by two lipids, that is hydrophilic head and hydrophobic tail, in which the aqueous structure, the drug is being enclosed. General structure of liposomes, it contains lipid bilayer in which water soluble drug is carried in hydrophilic region and water insoluble drug is carried in hydrophobic region. Now the compositions of liposomes. Components of liposomes combined with lecithin which is a mixture of phospholipids and cholesterol. Phospholipids, what are phospholipids? Phospholipids are fatty substance, the major structural component of cell wall and biological membranes. Phospholipids are amphipathic moieties with hydrophilic group which has hydrophilic head and hydrophobic tails. Two types of phospholipids exist, phosphodiglycerides and sphingolipids, together with their corresponding hydrolysis products. Phospholipids have phosphatidyl moiety which is known as tail with different head groups like choline, ethanolamine and serine. The most common phospholipid is phosphatidylcholine molecule. Phosphatidylcholine is a glycerol bridge which have hydrophobic acyl hydrocarbon chain having 10 to 24 carbon atoms with hydrophilic polar head group, as we can see in the structure. Phosphatidylcholine occurs in natural form which is amphipathic molecule which consists of hydrophilic polar head and glycerol bridge which is hydrophobic tail. The amphipathic molecule self-organized in ordered supramolecular structure when confronted with solvent. The most common natural phospholipid is phosphatidylcholine. Naturally occurring phospholipids are phosphatidylcholine, phosphatidylserine. Some phospholipids are also occur in synthetic form which are diasteroyl phosphatidylcholine, diasteroyl phosphatidylethanolamine and etc. Phase transition temperature, as it is very important temperature, due to at various temperatures, phospholipid membranes can exist in different phases. The transition from one phase to another can be detected by technique like microcalorimeter. This is due to the fatty acid chain adopting a new conformation other than all the trans straight chain configuration. The next component which is involved in the formation of liposomes is cholesterol. Cholesterol, it includes incorporation of sterols in liposomes bilayer, can bring about major changes. Cholesterol does not by itself form a bilayer structure, it acts as fluidity buffer. It is incorporated into phospholipid membrane in very high concentration up to 1:1 or even 2:1 molar ratio of phosphatidylcholine. The main aim of cholesterol is it increases the separation between the choline head groups and eliminates the normal electrostatic and hydrogen bonding interactions. Role of cholesterol in bilayer formation. As we have discussed, cholesterol mainly act as fluidity buffer. After interaction with phospholipid molecules, it alters the freedom of motion of carbon molecules in the acyl chain. It also restricts the formation of trans conformations. Now, what is the mechanism of formation of liposomes? As we have known, in aqueous media, phospholipids are not soluble and they closely align in planar bilayer sheets or lipid cakes, which are thermodynamically stable. In bilayer sheets, polar head groups face outwards into the aqueous medium. The lipidic chains turns inward to avoid the water phase, which forms a double layer. For the liposomes to be formed, it is then hydrated. The lipid cakes, that is lamella, swells eventually to form and they curve to form a closed vesicles in the form of spheres. These spheres are also known as liposomes. Amphipathic molecules other than phosphatidylcholine form micellar structure in the preference to bilayer sheet. In the structure, you can see the note we have discussed, bilayer sheet is formed due to which the formation of liposomes occur. Now, the main advantages of formation of liposomes, they are as liposomes increase the efficacy and therapeutic index of drug. It also increases the stability by encapsulation. It provides passive targeting to tumor tissues, flexibility to couples with site-specific ligands to achieve active targeting. Liposomes also improve the pharmacokinetic effects. The liposomes reduces the toxicity of encapsulated agents. It also provides the facilitation of transport across membranes, and these are naturally occurring lipids which are non-toxic and biodegradable. With some advantages, there are few disadvantages which are also related with liposomes, which are liposomes have low solubility in water, production cost is high, these have short half-life, sometimes the formation of liposome, that is phospholipid, undergoes oxidation and hydrolysis like reaction, leakage and fusion of encapsulated drug and molecules may occur. Now, the important parameter, which is classification. Liposomes has been classified in 6 type classes. On the basis of composition and application, liposomes are conventional liposomes, fusogenic liposomes, pH sensitive liposomes, cationic liposomes, immunoliposomes and long circulatory liposomes, which are called stealth liposomes. On the basis of structural parameters, that is their size, which is in micrometer or nanometer range, liposomes are divided as multilaminar vesicles, oligolaminar vesicles, unilaminar vesicles and etc. On the basis of liposome preparation, liposomes have been classified by vesicles prepared by reverse phase evaporation, multilaminar vesicles by reverse phase evaporation, stable plurilaminar vesicles, dried reconstituted vesicles and etc. These are some different type of liposomes which are varied on their size. Small unilaminar vesicles, large unilaminar vesicles and multilaminar vesicles. Liposomes and cell interaction. How does liposomes interact when it is incorporated in our body? 4 mechanisms are involved, which are adsorption, endocytosis, fusion and fusion of cell with vesicle and lipid exchange. Liposomes have been incorporated in bloodstream, which are then taken by reticuloendothelial system. Then the liposomes are engulfed by macrophages. The process is known as endocytosis. Then the phagosome and lysosome combine with each other and form phagolysozyme. And the membrane of phagolysozyme consists proton pumps which decreases the pH of phagolysozyme, and the enzyme phospholipase destruct the liposomal membrane and liposomes get interacted with the cells. Now the method of preparation and drug loading. How will you prepare liposomes? The general procedure involves 4 steps. Preparation of lipid for hydration, hydration with agitation, sizing for homogeneous distribution of vesicles, removal of non-encapsulated material. These are the method for the preparation of liposomes, general procedure, and the drug loading in the liposomes may be done by active loading or passive loading. Method of preparation. As we know, the method of preparations are categorized on the basis of lipid dispersion. The first method which is used for the preparation of liposomes is physical dispersion technique. It involves lipid film hydration by handshaking, non-handshaking method. Second one is high shear homogenization or sonication. Third method is membrane extrusion. Next is microfluidizer techniques for microemulsification. Next one, French pressure cell, then dried reconstituted vesicles and fusion method. These all methods were in physical dispersion technique. The second technique used for the preparation of liposomes are solvent dispersion techniques, which involves ethanol injection, ether injection, reverse phase evaporation vesicles, and emulsion and double emulsion vesicles. The third technique which can be used for the preparation of liposomes is detergent solubilization technique. Now we will explain all the methods in detail. So first technique was physical dispersion technique. Liposomes are produced when the thin lipid films are hydrated and stacks of liquid crystalline bilayer becomes fluid and swell. Then the hydrated lipid sheets cut off from lipid film during agitation and self-close to form LMV, that is large multilaminar vesicles. LMV, large multilaminar vesicles, particle size is reduction by sonication or extrusion. In this, 2 steps are being followed, that is first lipid film hydration by handshaking or non-handshaking, that is the first method. It involves 2 steps. The step 1 is preparation of film for hydration. In this, lipid and organic solvent which can be chloroform and methanol are taken and lipid is mixed thoroughly in the solution. Then the solvent is removed. For small volume of solvent, dry nitrogen and argon stream is used. As if the volume of solvent is large, the rotatory evaporation method is used. Then the lipid film is dried to eliminate all the organic solvent by placing it in vacuum for overnight. And the lipid film can also be prepared by freezing it in dry ice or dry ice acetone alcohol bath. The frozen lipid cake is placed in vacuum pump and lyophilized until it is dried. Dried films are placed in closely tied suitable container. Then the second step involves hydration of lipid film. The dried lipid film can be hydrated by addition of aqueous medium, which followed by agitation for 1 hour and overnight stand. The temperature of hydrating medium should be above transition temperature. The hydration medium which can be included for the step 2 are buffer solution, saline, 5% dextrose solution or 10% sucrose solution. The obtained product are large multilaminar vesicles which is formed in suspension is downsized. This is the structure of the method we have been discussed. Now the second method is high shear homogenization or sonication. In this, multi-laminar vesicles are exposed to ultrasonic irradiation for producing small vesicles. This is done by using two types of sonicator, which are probe sonicator or bath sonicator. Probe sonicator is used for the dispersion, which require high energy in small volume, while bath sonicator is used for the large volume of dilute lipids. In this, the multilaminar vesicles are sonicated for 5 to 10 minutes, which are then converted into hazy transparent solution. Then it is centrifuged for 30 minutes and a clear small unilamellar vesicles dispersion is being formed. The main disadvantage of this method is probe sonicator deliver high energy and heat, which can cause lipid degradation, and also it can release titanium particles into the lipid suspension, which have to be removed. The lipid keynote is the lipid solution of large multilaminar vesicles clarified to yield small unilaminar vesicles of 15 to 50 nanometer. Next method is membrane extrusion. It is used for the preparation of large unilaminar vesicles and multilaminar vesicles. In this, the size of liposome is reduced by passing them through polycarbonate membrane. Before extrusion, large multilaminar vesicles are disrupted by freeze-thaw cycles and pre-filtering through large pore size. Filter with 100 nanometer pores yield large unilaminar vesicles of 120 to 140 nanometer. Next method is microfluidizer technique for microemulsification. This method is used to prepare small unilaminar vesicles and multilaminar vesicles from concentrated lipid dispersion. The lipid are introduced into fluidizer as a dispersion of large multilaminar vesicles or a slurry of unhydrated lipids in organic medium. In this, the microfluidizer pumps fluid at very high pressure through a orifice which is 5 micrometer in range. Then it forces along defined microchannels which direct two streams of fluid to collide together at right angles at very high velocity, thereby affecting an efficient transfer of energy. The fluid collected is recycled through the pump and interaction chamber until vesicles of spherical dimensions are obtained. The advantage associated with microfluidizer is sample with high percentage of lipids can be easily treated. Next technique is French pressure cell. This technique was invented by Charles Stacey French. In this technique, large vesicles are converted to small vesicles under very high pressure. This technique yields or oligolamellar liposomes of intermediate size, which can range from 25 to 75 nanometer in diameter depending on applied pressure. These liposomes are more stable as compared to sonicated liposomes. These are suitable for drugs and compounds which can be degraded by ultrasonic radiations. Next method for preparation is dry reconstituted vesicles. It involves freeze drying the dispersion of empty small unilamellar vesicles followed by rehydration with aqueous fluid which have material to be entrapped. It is useful for the preparation of small unilamellar and oligolaminar vesicles. Advantage associated is it can provide high entrapment of water soluble components and bioactives. Next method is fusion method. This method protects lipid and entrapped material from harmful physicochemical environment. French fusion method is used for the preparation of unilamellar vesicles in large quantities. Fusogenic agents are used for fusion of small unilaminar vesicles for increasing entrapment efficiency. Alteration in pH can increase surface charge density of lipid bilayer and bring on spontaneous vesiculation. The next technique involved is solvent dispersion technique. In this process, lipid solution in organic phase is brought in contact with aqueous phase, including material to be entrapped inside the liposomes. Solvent dispersion technique includes various methods, in which the first method is ethanol injection. Ethanol injection, it is suitable for incorporating hydrophobic and hydrophilic drugs into the liposomes. It is suitable for the preparation of small and large unilaminar vesicles. In this method, the ethanol along with lipid is injected with 22 gauge fine needle and rapidly with the excess of saline and aqueous medium. The rate of injection should be suitable to attain absolute and rapid mixing. Next method is de-emulsification method. In this, generally we know liposomes are prepared in two steps. First is inner leaflet of bilayer, then the outer half. In this method, the aqueous medium and material to be entrapped is mixed together in a high volume of organic solvent of lipid, agitated mechanically, due to which the breakdown of medium to water droplets occur, which results in the formation of water in oil emulsion, and it is stabilized by phospholipid monolayer. Next method is double emulsion method. In this, the organic solution which already contains water droplets, that is water in oil, is introduced into excess aqueous medium, followed by mechanical dispersion causing phase inversion, that is WO, water in oil emulsion is being converted into OW emulsion. The multiple emulsion, that is WOW vesicle, is formed by double emulsion method, two aqueous compartments being separated from each other by two phospholipid monolayer. The hydrophobic surface of monolayer face each other with a thin film of organic solvent. Removal of organic solvent results in the formation of unilaminar vesicles. Next method is rapid solvent exchange method. In this method, the organic solvent vaporizes due to vacuum before contacting aqueous phase. The lipid mixture then precipitates very quickly in aqueous buffer solution, forming liposomes. Next method is reverse phase evaporation. In this method, both the phases, that is aqueous phase and organic phase, are subjected to bath sonication. Droplets are then formed which are dried to semi-solid gel in a rotary evaporator under reduced pressure. In this stage, monolayer of phospholipids surrounded by water compartment. After that, mechanical shaking with vortex shaker collapses few water droplets and lipid monolayers. Collapsed lipid monolayers form outer layer of stable vesicles, thus forming bilayer of small unilamellar vesicles. Next technique involved is detergent solubilization technique. In this technique, it is associated with phospholipid molecules and serve to the screen the hydrophobic portion of molecules from water. The structures formed as a result of this association is known as micelles. The concentration of detergent in water at which micelles instantly form is called critical micellar concentration or CMC. Liposome size and shape depend on chemical nature of detergent, concentration and other lipid involved. The methods used for the removal of detergents are dialysis and column chromatography. Hydrophilic non-bilayer interact with the low molecular weight compounds have low entrapment. This method is suitable for the preparation of immunoliposomes.